Welcome to the solutions video for problem set 10 for Kevin Ahern's BB450 class at Oregon State University. In this problem set, I'll be going through dealing mostly with the sugars relating to nomenclature and structure. The first question asks students to categorize the following sugars as epimers, anomers, enantiomers, diastereomers, or none of the above. The first pair, A, consists of alpha-D-glucose and beta-D-glucose. And of course, the prefixes in front, the Greek letters in front, indicate that these are two anomeric forms of the same sugar. So these two sugars, of course, are anomers. The second pair, D-glucose and L-glucose, refer to the fact that the D and the L, you recall, when given the same name of a sugar, refer to different mirror images of the two. And mirror images of sugars, of course, are known as enantiomers. The next pair, glucose and fructose, we remember that glucose is an aldose and fructose is a ketose, meaning that glucose has an aldehyde group at the end and fructose has a ketone group within it. Now, neither of these aldose or ketoses have any names that fit above, epimers, anomers, enantiomers, diastereomers, at all. So it means that these two sugars, which are actually aldose-ketose pairs of each other, fit into the category of the names above as none of the above. The next pair, alpha-D-glucose and alpha-D-galactose, differ from each other only in the configuration of carbon number 4, meaning that they only have one carbon whose configuration is different between the two. These, of course, are known as epimers. The next pair, alpha-D-glucose and alpha-D-gulose, was a pair that I showed in the class presentation. These two sugars actually differ in the configuration of two different carbons. Now, when we have things that differ in configuration of more than one carbon, and they have the same chemistry, meaning that they're both aldoses, and they have the same number of carbons, which both of these do, then that means that these two are diastereomers. So diastereomers are sugars that differ in configuration of more than one carbon, have the same chemistry, and the same number of carbons. The last pair, alpha-D-glucose and alpha-D-ribose, don't have the same number of carbons. So we really can't compare the two, and these two would also fit into the category of none of the above. Question number 2 asks, if you oxidize carbon number 1 of glucose, you make gluconic acid. If you oxidize carbon number 6 of glucose, you make glucuronic acid. Both of these compounds can ionize, meaning that they can lose the hydrogen off of the carboxyl group of each of the two. Both of these compounds, therefore, are polar, and both of these compounds are very soluble in water because of their ability to ionize. Glucuronic acid, though, can do one thing that gluconic acid cannot. What is that? Well, let's look at what those structures are to understand that a little bit better. Glucuronic acid looks something like this in the alpha configuration. Let's put it in the beta configuration, looking like this. Notice the carboxyl group on carbon number 6 is characteristic of glucuronic acid. Gluconic acid, on the other hand, has the carboxyl group on carbon number 1. So this is the structure of the two carbons that I have in the problem. The question then says, glucuronic acid can do one thing that gluconic acid cannot. What is that? Well, you may remember when I said in class that whenever we alter the hydroxyl on the anomeric carbon, that locks the carbon into position, meaning that it can no longer flip between the alpha and the beta configurations. And that's because the anomeric hydroxyl is necessary for the sugar to go back into the linear form. So this sugar, glucuronic acid, which has the free hydroxyl on the anomeric carbon, can go back to the linear form and can change beta to alpha, alpha to beta. This sugar can't do either. Again, this sugar can't do either, and that's because it has an altered hydroxyl group on the anomeric carbon. Question number 3 says that when a reducing sugar reacts, what happens to it? Well, this is important to remember. A reducing sugar is called a reducing sugar because it is readily oxidized. And it is a reducing sugar in that it is reducing something else. So a reducing sugar gets its name from the fact that it gives up electrons to something else. And in giving up those electrons, it is reducing something, which is why it's called a reducing sugar. But in the process of giving up its electrons, a reducing sugar is oxidized. The last question says, why does the Fischer form of a sugar not have an anomer? And the answer to this question is pretty straightforward. You may recall that the cyclization reaction is what caused the anomeric carbon to be created. We look, for example, at glucose. Glucose has this configuration. In the Fischer form, 1, 2, 3, 4, 5, 6, where the hydroxyls are indicated by the bars on either side of the sugar. Notice that carbon number 1, which is right here, does not have any asymmetry because it only has 3 things attached to it: a hydrogen, a double bonded oxygen, and another carbon. If, however, we make a ring form of that same sugar, what we create is something that looks like this. Now, in this case, we see that this carbon, number 1, has a hydroxyl on it where it had a double bonded oxygen on it before, and this carbon now has 4 things attached to it. And I can label a hydrogen here. It's bounded to an oxygen, it's bounded to a hydroxyl, and it's bonded to the rest of the ring, meaning that this is a new anomeric carbon. This straight chain form only has 3 things bound to it and cannot exist in a stereoisomer, therefore is not an anomeric carbon.